I'm not Rebecca Neumann. My name is Greg Miller. I'm the current interim dean of the College of Engineering, but also formerly chair of civil environmental engineering. And so one of the first things I did when I took that position was I hired Rebecca. So it's wonderful to be able to introduce her to you. This is our series on the future of food. Try to say future of food 10 times fast. It's harder than you might think. But I only had to say it once, which is very interesting because clearly food is something that we all deal with every day. It's also interesting that earlier this week, in fact, yesterday, one of my coworkers mentioned that she was having some work done with a pump in her well, and one of the things that they were concerned about was arsenic in the water. And so I said, are you going to the lecture? She said, oh, really? What? Anyway, she's going to watch the video. So these are very timely and important topics. And Professor Neumann works in the area of hydrobiogeochemistry. And it turns out that if you think about it's not like her curriculum, right? The classes you're taking today, but if you think about what it is that happens between water, food, the soil, you get that laundry list of things that work as a system. And so that's the domain in which she works. The temptation always is to tell you what I think about what she does, but you're here to listen to her and not me, so I won't go very far down that road. She's a wonderful colleague, and she did her PhD at MIT and then a postdoc at Harvard, and then we were able to extract her and her husband and one kid at the time, but now two, and bring them to Seattle, where she's, again, a faculty member for us. But for now, it's my pleasure to introduce Professor Rebecca Neumann. Okay.
So thank you, Greg, for the kind introduction. I want to thank the College of Engineering for putting this lecture series together on the future of food and inviting me to be part of it. I hope that many of you were able to come to the first talk in this series, which was given by my colleague, Faisal Hossein. He gave a really interesting and very inspiring talk about tools he's developing and actually delivering to the farmers in Asia to help them grow more food with less water. Tonight, I'm going to shift the conversation and talk to you about arsenic, because arsenic makes its way into our food systems, but also into our water and into the ecosystems that surround us. In my talk tonight, I'm going to tell you about arsenic and why you should care about it. And on a fundamental level, I think everyone should care about arsenic because it is a carcinogenic contaminant and it has negative impacts on human and ecosystem health. But for those of us sitting here tonight, we should care about it because it's a local problem. So this is not something that's just affecting people off in far off distant parts of the world. It's something that is actually affecting the Puget Sound and something therefore we should all be concerned about. And that's also something I'm going to get into my talk tonight, is the local aspect of the contamination problem. And then finally, I want to let you know that I have brand new data that I'm showing to you tonight. So my collaborators and my colleagues have been working really, really hard to generate some new data so I could show it to you here tonight. So I'm really excited, and I think you're in for a real treat, or at least as much of a treat as a scientific talk can provide. So arsenic, it is a naturally occurring, again, carcinogenic contaminant. So we find it in rocks, soils, and sediments all around the world. It's naturally occurring, but there have been in the past some human activities that has released arsenic out into the environment and caused concentrations in these impacted areas to rise above what we'd consider to be background or baseline levels. And these activities include smelting and mining. So arsenic tends to be associated with ores that we find valuable, like copper. And so arsenic gets emitted from these processes more as an unwanted byproduct. There's also been past poor handling of industrial discharge, and then what people often find surprising is that we used to use arsenic as an herbicide, so we were purposefully applying it. Many of these activities are no longer occurring, but the contamination that got released from these activities remains in the environment and continues to have impacts. And so this is why we call it a legacy contaminant. So when arsenic makes its way into the human body, it causes arsenicosis, which manifests itself externally with these types of lesions that you find on palms of hands and bottoms of feet. And these lesions are quite visually graphic, and so they're often what gets shown. But fundamentally, it's all the damage that's caused on the inside of the body, that is the real problem. So here I have a slide showing all of the different systems in the human body that gets affected by arsenic. So our nervous system, impaired intellectual function, our cardiovascular system, heart disease, heart attacks, renal system, kidney cancer, bladder cancer, these skin lesions, skin cancer, which I showed in the other slide. Endocrine system results in diabetes. Lots of impacts on our respiratory system, tuberculosis, bronchitis, lung cancer, liver cancer, and then especially if children get exposed to arsenic at a young age, it can have some pretty negative effects, increased cancer risk as adults, infant mortality, neurological impairment, reduced birth weight. So lots of negative consequences, but again, this is from chronic continual exposure to arsenic, so not like a one-time dose of it. So when we find arsenic out in the environment, in the soils and in the sediments, it doesn't really pose too much of a health threat to us, except for in the case, which is most often of children who kind of accidentally consume that dirt. So some children are more likely to be eating dirt, for example, than adults are. Yes, usually. What we really worry about is when arsenic moves off of the soil and sediments and into the surrounding water. Because once we have arsenic in water, it can enter into our drinking water system, it can enter into our irrigation systems, it can be taken up by plants, and if we're growing food, that arsenic can get translocated up into the grains of the plants that we're growing, and it can enter in and affect our aquatic ecosystems. And so tonight, I'm going to talk about arsenic in each of these components in water, in food, and in aquatic ecosystems. So this movement of arsenic off of soil and sediments and into water is a process which is mediated by microbes out in the environment. And so this is microbial respiration. This is a redox reaction. And I know some of you are starting to feel nervous because it's been many years since you took chemistry. But I want you to know that you are actually quite familiar with this reaction. You're performing it right now as you sit here listening to me talk. We are in our bodies doing human respiration. So we take organic carbon, we oxidize it to CO2, and we need to couple that oxidation reaction with a reduction reaction, and we use oxygen. We reduce that down to water. So this is the redox reaction by which we get all of our energy to live. Microbes in the environment are just the same. They need energy to live, and so they're performing redox reactions. They take organic carbon that they can find in the environment, they oxidize it to CO2. But what happens is when oxygen in the environment runs out, instead of keeling over and dying, which is what we would do, they are more flexible. So they begin to look to other elements. And iron is a very common element. And this is because iron minerals are very prevalent out in the environment. And they'll take iron 3 in these minerals and reduce it down to iron 2. Iron 3 is a solid phase species of iron, and then iron 2 is a dissolved phase species of iron. So through this reduction reaction, they end up dissolving these iron minerals. Turns out that arsenic is usually associated with these iron minerals. So just conceptually, you can understand, any arsenic which is associated with the part of the mineral that's beginning to dissolve and release into the water, you're also going to be releasing that arsenic, right? So we call this reductive dissolution. So we're dissolving these minerals and we're releasing all of the arsenic which was associated with those minerals. And so it's through this redox reaction that microbes mobilize arsenic off soil and sediments and into the surrounding water. This redox reaction has had negative consequences on groundwater chemistry throughout the globe. So here I have a map showing the colored areas is the probability of finding arsenic at levels exceeding 10 micrograms per liter. So that's the drinking water standard right now for arsenic in the United States, as a result of this redox reaction. So you can see many places in the world being impacted by mobilization of arsenic off soils and sediments and into the groundwater. I want to draw your attention to Bangladesh and West Bengal because this part of the world has been severely impacted by this microbial reaction. And this is because groundwater is the primary source of drinking and irrigation water within this part of the world. So what happened is in the 1970s, many aid organizations and private donors came into Bangladesh and West Bengal, and their goal was to reduce the high rate of waterborne diseases. And so they did this actually successfully by moving the country off of contaminated surface water and onto groundwater. Unfortunately, after this mass conversion of the population onto groundwater occurred, it was discovered that the groundwater was severely contaminated with arsenic. So this meant that there was millions, more than 50 million people exposed to unsafe levels of arsenic in their drinking water. And just to put this in perspective, a researcher publishing in the Bulletin of the World Health Organization, I have him quoted here. He's just emphasizing that the scale of this environmental disaster, the number of people that are being affected by this contamination, was greater than any seen before, beyond famous accidents such as in Bhopal, India, and Chernobyl in Ukraine. So, a really large human health crisis in Bangladesh and West Bengal, and this motivated a lot of work into understanding how arsenic behaves in the environment and what the consequences of arsenic are on the human body. And so much of what we know today was largely motivated by this contamination problem. And just to visually drive that point home, here I have a graph showing arsenic publications normalized by a metric for all publications. So it's giving us an idea of the knowledge generation around arsenic over time. And we can see here in the 1980s, arsenic research was going along at some baseline level. The first case of arsenicosis that got documented in West Bengal happened here in 1983, but unfortunately, it did not get published until 1995. So nobody knew about it except for the person who documented it and maybe the small cohort of people that person talked to. What happened in 1995, this documented case got published, and then there was an international conference which was held to raise awareness about this contamination problem. And you can just see the number of publications resulting after that conference taking off. So there was just a real drive to advance understanding about arsenic. Here in 2001, the British Geological Survey, they did a countrywide survey through Bangladesh, finding every well that they could and measuring the arsenic concentration in those wells. And based on this nationwide survey, they estimated that there was 57 million people drinking water with arsenic exceeding 10 micrograms per liter. And then after that point in time, you can just see publications begin to skyrocket. I was fortunate enough to be one of the scientists that got to help work on this problem. I'm highlighting here the 3 years where I traveled to Bangladesh and conducted field work as part of my PhD research. And I was focused on trying to understand the source of this organic carbon, which was fueling the microbial reaction that I stepped you through, which is responsible for mobilizing arsenic. So publications have kind of plateaued and started to drop here over time, but we still continue to learn things. For example, in 2010, a really shocking publication came out which demonstrated that arsenic in drinking water in Bangladesh increased the rate of death by 34 to 68%. So a really negative health outcome just from chronic exposure to arsenic in drinking water. Okay, so Bangladesh, West Bengal raised a lot of awareness about the possibility of arsenic in groundwater and really motivated a lot of research into understanding how arsenic behaves. At this time, though, we also started to think about the possibility of arsenic making its way into the food system. And this is because in Bangladesh, as I mentioned, groundwater is the primary source also of irrigation water. So farmers in this part of the world, they grow a dry season rice crop and it gets irrigated with groundwater, but this groundwater has arsenic in it. So this means that every year when the farmers are irrigating their fields, they're adding a little bit more of arsenic onto that soil every year. And so researchers started to wonder, well, what does that mean? What does it mean if we're adding arsenic and increasing arsenic concentrations in our soils? So in general, this is one of many studies coming out of Bangladesh. In general, when arsenic in soil increases, we tend to see increasing concentrations in the grain of the rice plants grown within that soil. This is not always the case, but most of the time this is what we see. And then what was more surprising, a publication coming out around the same time, is that we actually saw a decrease in grain yields. So as soil arsenic is increasing, we're seeing a drop in the quantity of rice which is being grown. So not only is arsenic bad for the human body, it's also bad for the rice plants. So basically the rice plants are struggling when they get exposed to arsenic, and they're just not as healthy, and so they're not producing as much rice grain. So evidence from Bangladesh that this contamination of soils with arsenic negatively affects both the quality and the quantity of rice which we're growing. So this was focused on Bangladesh. Researchers started thinking, well, what about the rest of the world? Rice is grown in more places than just in Bangladesh and West Bengal. And so these researchers, they went around and bought polished white rice from a number of different marketplaces all around the world, and they looked at the arsenic content of that rice which they collected. Here I'm showing you data from their study just for Bangladesh in the United States. They went to a lot of different places. And this top panel is the model global distribution, so a synthesis kind of all the data that they collected from all the different countries. Okay, well, surprising. Rice grown in the United States has more arsenic in it than rice grown in Bangladesh, right? So I just was telling you, well, they're irrigating their fields with arsenic-contaminated groundwater, and yet we're growing rice in the United States that has more arsenic in it than the rice which is grown in Bangladesh. It turns out that in the United States, in the southeastern part of the US where we grow most of our rice, we're growing on former cotton fields, and we used to use arsenic-based herbicides on these cotton fields. So this is an example of that legacy arsenic contamination coming back and having modern-day consequences for our food system. Okay, so what do these levels of arsenic in rice mean? So unfortunately, there is no standard for arsenic in food. So in drinking water, we have a drinking water limit. 10 micrograms per liter is the standard that's currently set here in the United States. If things are above that, it's considered unsafe, and if we're below that, it's considered safe. Food doesn't have anything like that, so the best we can do is kind of deal with rough guidelines and thresholds based on results from academic studies. So in 2013, another pretty foundational study came out. This was the first one to connect arsenic in rice directly with a human health effect. So it's always that kind of direct connection, I think, which is hard to make when we're trying to say X causes Y. So this is one of the first studies that was able to do that for rice arsenic. And what they showed is that when you exceeded a concentration of 0.2 milligrams per kilogram of arsenic within the rice, you started seeing genotoxic effects. So that basically means that the DNA got damaged. And when we have damaged DNA, we get mutations to our cells, and then cell mutations can lead to cancer. So it's basically like a first step on the road to cancer. So that threshold is at 0.2 milligrams per kilogram. And then more recently, in 2016, the Food and Drug Administration finally came out with a guideline, but it's pretty narrow. So this is regulating, or it's not even regulating, it's just a guideline for infant rice cereals. Okay, so a pretty narrow set of things that we're thinking about, but nonetheless, it's a guideline, and they're saying we should not exceed 0.1 milligram per kilogram of arsenic in infant rice cereals. So when we look at these kind of thresholds, we see, well, much of the rice in the world is below this genotoxic threshold. A lot of it is, at or slightly above this guideline. Of course, in the United States, our rice is well above this genotoxic threshold. So work that I've been doing here at the University of Washington has been thinking about how is this issue of arsenic contamination of rice going to change as we move into the future. And we have an altered climate system. So here on this map, I'm showing average regional rice output. Not surprisingly, most of the rice in the world is being grown in South and Southeast Asia. And then next to this, I'm showing predicted changes in average surface temperature. This is resulting from the most recent IPCC report. This top panel is kind of the best case scenario, and this lower panel is our worst case scenario. And what this is showing us is that in these rice growing regions, we can expect surface temperatures to increase anywhere from around 1, up to 5 degrees Celsius. So we're moving into a warmer future. Well, why should we care about warmer temperatures? Microbial reactions, they get faster when temperatures get warmer. So the hypothesis that we had when we moved into this research project was that under warmer temperatures, this microbial reaction is going to speed up, and so we're going to get more movement of arsenic off of the rice soils and into the soil water. So if we have more arsenic in the soil water, this means we can get more arsenic being taken up by the roots of the plants, more translocation of that arsenic into the above ground biomass and into the grain of the rice plants. So to test this hypothesis, graduate student in my group, Yasmin Farhat, along with the help of multiple different undergraduate researchers, conducted an experiment. So she grew rice plants under 4 different temperatures. And then once they came to maturity and the grains had fully ripened, she basically collected and harvested these rice plants and analyzed the arsenic content in the tissues of the plants. And these data are weeks old, so she worked so hard to get these data to me in time so I could show them to you tonight. So this is exciting. What I'm showing you is average stem arsenic concentrations and then average grain arsenic concentrations as a function of the temperature at which the plants were grown. And so what we can see is a very clear, strong, positive correlation between levels of arsenic in these above ground tissues and the temperatures at which the plants were growing. You might be noticing that we seem to be missing this top data point, so grain from the plant grown under our warmest temperature. Well, that plant was so heat stressed, it just didn't produce any grain. So the yield was 0, there was nothing there for us to analyze. We can compare our measured levels to these thresholds and guidelines, which I presented previously. We're working here with a pretty low arsenic system, so our soils were not contaminated with arsenic. So overall, we had a pretty arsenic-free system. So down in these lower temperatures, the grain that we were producing was below this FDA guideline, well below this genotoxic threshold. But nonetheless, what we're seeing, right, is even when we have a low arsenic system, if we start marching the temperature up, we can start moving concentrations up into these regions where we begin to start having some concerns. So unfortunately, the results of our experiment are suggesting that this issue of arsenic contamination in rice is going to get exacerbated as we move into warmer climate conditions. Okay, lots of bad news. You might be starting to worry about all the rice that you ate in the past, wondering if you'll ever eat rice again, I want to point you towards at least some concrete advice to help you deal with maybe some of that nervous feelings you have. So Consumer Reports came out in 2012 and 2014 with really two nice studies. They did a comprehensive review of arsenic in rice and a range of rice-based products, and they've also started to look at other grains like quinoa, for example. And in their reports, they provide some really good take-home messages for consumers. One of them is they identify the types of rice that are low in arsenic. So these include basmati rice, grown in India, Pakistan, and California, and then sushi rice grown in the United States. So next time you're at the grocery store, you want to buy rice, focus on these varieties. They're the ones with the lowest levels of arsenic. They also highlight the rice that has the most arsenic in it. So avoid, if possible, rice from Texas, Louisiana, and Arkansas. So these regions where we have grown rice on fields where arsenic-based pesticides were used in the past. And then in general, regardless of the variety, brown rice has more arsenic in it than white rice. And this is because the arsenic actually gets deposited on the outer part of the rice grain, so the part that makes the rice brown. And so when they polish that rice and make it into white rice, that outer sheath gets taken off, so it brings down the levels of arsenic. So if you have a choice between brown rice and white rice, go for the white rice. Okay. I know it's opposite of what you maybe thought. You thought, oh, brown rice is so healthy, I'm getting whole grains. But and then I think a really, really nice outcome of the report is that they actually developed a point system. So they say aim for no more than 7 points a week, and then they give you this table where they outline, for different rice-based products, for a given serving, how many points is that for a child versus an adult, okay? And this is just a snippet of their table. It's quite long and extensive. So I would say, if you like to eat rice, if you're gluten-free and you just inevitably end up eating a lot of rice, this would be a good table to kind of look at and gauge and try to just make sure you're staying in safe levels. Okay, so we've been talking about arsenic in water, groundwater, arsenic in our food system. We've been thinking kind of globally and South and Southeast Asia. So what I want to do now is bring the discussion local and talk about arsenic in aquatic ecosystems. So it turns out that here in the Puget Sound region, our landscape is contaminated with arsenic, and this is because the Asarco smelter operated for just under 100 years outside of Tacoma, Washington, and this was a copper smelter. Arsenic got released out of the smokestack just as an unwanted byproduct. It blew across the landscape, settled down, entered into our soils and into the sediments of our lakes. Washington Department of Ecology has a really nice program which is called Dirt Alert. And this is a snapshot just of the website. And here they show you measured concentrations of arsenic in soils throughout the region. So darker brown color is higher levels of arsenic, and then you move out into lower levels of arsenic. And the nice thing is you can actually go to this website and type your address in. So if you're worried about levels of arsenic in the soils around your home, you can type your address in, and it will take you to that location. So I've just picked some place in Gig Harbor, which is kind of in the heart of the plume, and they can tell you, what the expected concentration of arsenic is in soil near your property. They let you know if your property is inside or outside of the smelter plume. And then they also give advice on simple, healthy actions you can take to reduce your arsenic exposure from this contaminated soil. So again, this is soil, so you're mostly just worried about that soil making it into your mouth. So they say wash your hands, keep your shoes outside of your house, mop up your floors, and wash off toys that maybe have dirt on it. So some real kind of easy, straightforward ways to limit your exposure to the contaminated soils. The Dirt Alert program is really great, but it's only focused on dirt. So an area that's been largely overlooked are the lakes in our region. So we know that the sediments of our lakes have also received this arsenic contamination from the Arsenic smelter. My colleague Jim Gowell down at UW-Tacoma, he went around to a bunch of lakes here in the Puget Sound lowland, collected sediment out of the lakes and measured the arsenic content. And those are the data I'm showing you here. Each of these dots is one of the studied lakes, and the size of the circle lets you know how much arsenic was in that lake. So the bigger the circle, the more arsenic. So we see that there are lakes all around this region which are contaminated with arsenic, and some of them extremely contaminated with arsenic. In general, I would say that there is a lack of understanding of how arsenic in these lake systems affects human and ecosystem health. And so that's a gap that I and a whole number of other researchers have been trying to fill. We're on a large collaborative project trying to understand arsenic mobility, bioaccumulation, ecotoxicity within these lake systems. There's lots of people in this project. I'm showing you just the people who were involved in the data that I'm showing you tonight. So there's a team of us in the College of Engineering. I've lumped my dad in as an honorary member. He came into the lab and taught me how to dissect fish so that I have fish data to show you tonight. So we can thank him for that. We have colleagues over in the College of the Environment and a number of people down here at UW-Tacoma. Okay, so when we're thinking about arsenic in lakes, the story is very similar to the story that I've been telling all night long. We have arsenic in these sediments. It's on the solid phase, but we have microbes out there in the sediments. Those microbes mobilize that arsenic off of the sediment and into the surrounding water within the lake bed. Once we have arsenic in the water of the sediments, it can get transported up into the overlying lake water. Once we have arsenic in the overlying lake water, it can then get taken up into phytoplankton. So phytoplankton mistakenly take arsenic up out of the water. They're really trying to get phosphorus out of the water. It's one of their needed nutrients, but phosphorus and arsenic are chemically very similar. So they take it up by mistake, thinking it's something they need, and instead it's something that's bad for them. The zooplankton eat the phytoplankton, get exposed to arsenic, and then the fish eat the zooplankton. Of course, once we have arsenic in fish, we have the possibility of arsenic making its way into humans. So we've been studying these lakes for a number of years, and we've had some pretty surprising results. Notably, arsenic that we're finding in this aquatic food web is not directly related to the amount of arsenic that we're seeing in the lake bed sediments, or even the maximum concentrations of arsenic that we're finding in the overlying lake water. So what I want to do now is step you through our results and help explain why we're seeing this pretty surprising and unintuitive result. So we're going to focus on two lakes, Angle Lake and Lake Killarney. These are two of the most impacted lakes in the region. They each have around 200 milligrams per kilogram of arsenic within their sediments, so severely contaminated. When we look at the maximum levels of arsenic making its way into the overlying lake water, we find that both of them have very similar levels of arsenic, around 50 micrograms per liter, and these are maximum concentrations. But when we look at the phytoplankton, we see a huge difference in the concentrations of arsenic but in the phytoplankton between these two lakes. So I want you to note this is on a log scale, so maybe visually it doesn't look that different, but it is quite different, and this is why I'm giving you the actual numbers here. Okay, so same sediment arsenic, same maximum water arsenic, but dramatically different amounts of arsenic being taken up by the phytoplankton. What the heck is happening? Well, it turns out that Angle Lake is a deep lake, and Lake Killarney is a shallow lake. So why does this matter? It turns out that in Angle Lake, these deep lakes, these bottom waters often become stagnant and anaerobic. So this means that there's no longer oxygen inside of them. Aquatic life needs oxygen to survive. So that means that the aquatic life is hanging out at the top of the lake, and the arsenic is getting mobilized down here at the bottom of the lake. Okay, so we get spatial separation between where the arsenic is and where the aquatic life resides in these deep lakes. In the shallow lake, we don't have these deep bottom waters that become anaerobic. So when we're getting arsenic mobilized up out of the sediments, it's actually coinciding with waters that have oxygen in it, and so we actually get overlap between the contamination and the aquatic life. So we were thinking, well, maybe it's not maximum levels of arsenic that we should be looking at, but instead we should be looking at arsenic within oxygenated waters. So here I'm showing you data from 4 different study lakes, and I'm plotting concentrations measured in phytoplankton against the average concentration of arsenic measured in waters that contained oxygen. The circles are snapshots in time. So every time we went out to a lake, collected a sample of phytoplankton and collected our water samples. And then the diamonds are showing the average for the summer season. And so what we see is that arsenic bioavailability, or uptake of arsenic into phytoplankton, is related to the amount of arsenic that we're seeing in oxic waters. And this is noteworthy because water quality guidelines are currently focused just on these maximum levels of arsenic. But what our data are suggesting is that we should maybe not be looking at maximum levels when we're thinking about water quality guidelines. These guidelines are set to protect the health of the aquatic ecosystem. But instead of thinking about maximums, we should be thinking about levels of arsenic in waters that have oxygen, because these are the waters that are going to be housing the aquatic organisms that we're trying to protect. Okay, so if we come back now to our 2 study lakes, Angle Lake and Lake Killarney, instead of maximum levels, I'm now plotting levels of arsenic within oxic waters. And so you can see that we have almost an order of magnitude difference in the average concentration of arsenic within oxic waters of these 2 lakes. So now when I throw up the phytoplankton data, which you've already seen, it makes a little bit more sense, right? We're getting now this increased transfer of arsenic from this oxic water arsenic into the phytoplankton, right? So now what happens when we move up a trophic level? When we look at zooplankton, we continue to see a difference in the levels between the 2 lakes. You'll notice that there was a drop in concentration. So arsenic is being actively excreted out of the bodies of the zooplankton. So they're trying to get arsenic out of their bodies. And so that's why we're seeing less arsenic in those organisms than we are within the phytoplankton. But nonetheless, we're getting transfer of arsenic up the trophic food web. And then, yes, the new data, the fish data. We worked so hard to get it for you. When we look at fish, we continue to see this difference in arsenic. On average, the fish in Lake Killarney had 3 milligrams per kilogram arsenic in them, and the fish from Angle Lake had 0.1 milligram per kilogram. So we are seeing movement of arsenic through the aquatic food web all the way up to fish, which has those implications for people if they're going to be catching and eating fish out of these lakes, which people do. Again, there's no guidelines for arsenic in food, so the best I can do is compare these levels to average values collected from a synthesis study, which was published in 2006, where they looked at arsenic levels within fish across 17 contaminated sites. And so this is the average from that study. The fish in the average for fish in Lake Killarney is greater than that synthesis average, and then in Angle Lake, we're near that synthesis average. So what these data, I think, are indicating is that this shallow lake system, so Lake Killarney, where we're getting arsenic in oxic waters, these shallow systems appear to be more vulnerable to the same levels of contamination within the sediments relative to these deeper lake systems. So again, same levels of arsenic in the sediments of these two lakes, but because we're getting arsenic moving up into these oxygenated waters, we're seeing large amounts of arsenic being moved through the aquatic food web. Okay, so let's now zoom back out and take the global perspective. Here we are, Puget Sound. We are not the only place that is struggling with arsenic contamination in our lakes. So this map shows all of the lakes around the world that I could dig out of the academic literature, which were reported to be impacted with arsenic based on a number of activities. So northeastern United States, southeastern United States, we have lakes with arsenic from herbicide application. A lot of lakes here in Canada impacted from mining and smelting. And then more of an emerging problem coming out here in Asia, particularly in China. Industrial development has been going quickly in this part of the world, and there's not always been good handling of that industrial waste. And so we're seeing kind of the emergence of arsenic making its way into lakes in that part of the world. So this map is here to show you that arsenic contamination of lakes is a global problem. You might be looking at this map and thinking, wow, most of the contaminated lakes are in North America. Don't make that assumption. This just simply reflects the fact that most of the scientists have been working on lakes in North America. So really, it's a call to action to be looking at lakes in other parts of the world outside of North America. And then one final thing is that most of the lakes which are represented here are large, deep, famous lakes. Right? So it makes sense. If you were a scientist, you want to study something that matters, you're intuitively going to be drawn towards the big famous lakes, like Lake Washington, right? But in reality, what our results are suggesting is that it's not necessarily these big famous lakes, which are also deep, but it might be these really small, shallow lakes, which are the most vulnerable to this contamination. And here I just have a satellite overview of Lake Killarney, just demonstrating how small it actually is. And you'll also notice it's embedded in a highly urbanized area. So this lake is serving an important kind of urban ecological function, and it is heavily used by the people that live in this area. So kind of another call to action that we shouldn't be overlooking, our small kind of urban lakes and ponds. Okay, so I took you through a lot. Time to bring it all back together again. If you go home tonight and you only remember one thing, you should remember that arsenic is important, and it's a carcinogenic contaminant, and it impacts our food, our water, our plants, and our animals, both locally here in the Puget Sound and globally. Historically, the contamination crisis in Bangladesh and West Bengal really spurred and motivated understanding into how arsenic behaves in the environment and what the consequences are for humans exposed to arsenic. Unfortunately, this occurred with detrimental health consequences for the people living there. We have an understanding that arsenic affects not only the quality, but can also negatively affect the quantity of the food that we grow. And work coming out of my group is suggesting that this contamination problem is likely going to get exacerbated under a warmer climate. Nonetheless, there is concrete advice that exists for reducing your own personal exposure to arsenic. You can harness those nice points from the consumer reports. And then finally, arsenic contamination affects local and global aquatic ecosystems. Our work is indicating that the shallow lake systems might be more vulnerable to the contamination than larger deep lakes. There are consequences for this for both the ecosystems and the humans. that recreate and fish and eat fish out of these lakes. And then finally, a nice management outcome of our work is that we should maybe not just be looking at maximum levels of arsenic in our aquatic ecosystems, but be focusing on levels in oxygenated waters where the aquatic organisms are living. And with that, I want to just acknowledge the 3 primary sources of funding which have helped generate all of the work and data that I talked about tonight. And if you're interested, I keep a pretty up-to-date website, and I'm only about a decade behind the times, but I am finally on Twitter. So if that's something you do, you're welcome to, you can follow me on Twitter. Okay, that was that. I'm happy to take questions.